Hello, my name is Riddhimaan Tejas, and I'm a freshman at William Mason High School.
Hi, I'm Tejas Chen, and I'm a ninth grader at William Mason High School.
Our Earth is a beautiful place. However, humans have induced many problems into our Earth, some really small, others really large. One of the largest problems impacting our Earth right now is climate change. And there is another problem equally as large, but remains largely understudied, and that is microplastics.
According to Nature.com, the annual usage of microplastics has risen by 12.5 million tons over the past 70 years. Microplastics are small fragments of plastics that are less than 5 millimeters in size.
In order to tackle both climate change and microplastics, we decided to do a dual problem solving approach, and we discovered that plants are the solution. So what our project is we tested the effects of microplastics, a common microplastic called polyethylene, on the growth of high photosynthesizing plants.
So what is the purpose of our project?
The significance of our project lies in addressing an largely overlooked aspect of microplastic pollution, its potential impact on agricultural and food security. Our project is assessing how microplastic exposure affects plant growth, including measuring height, root development, number of leaves, and overall health, determining whether the presence of polyethylene in soil leads to differences in plant figure, as well as providing evidence that can inform future research and practical applications, especially in underdeveloped areas. What issue does our project address?
By focusing on the most commonly found microplastic polyethylene, on the growth of high photosynthesizing houseplants such as lettuce, radish, and mustard, our experiment aims to fill that knowledge gap, providing important data on how microplastic contamination may affect essential crops that are critical to the human food supply and carbon dioxide atmospheric removal. So how do different concentrations of polyethylene in soil impact the germination rate, growth, and overall health of common high photosynthesizing plants? That was our research question.
What did we hypothesize?
If varying concentrations of microplastics are added to the soil, plant growth would negatively be affected. Compared to control plants grown in microplastic-free soil, experimental plants will show reduced germination rates, shorter average plant heights and widths, stunted root lengths, and a lower number of leaves with diminished health. So what did we need for our experiment?
We needed some basic things that were taken from art stores as well as our houses, such as pencils, erasers, sharpies, as well as aluminum trays and trowels. However, some things did need to be bought from chemical stores like the microplastics. How did we conduct our experiment?
To perform the experiment, we had to use a control environment, so this took place in a house basement. We put the soil into 3 pots for the experimental and 3 pots for the control, and we used a scale to measure 1,210 grams of soil for each pot. Next, for each experimental group, we measured the appropriate amount of polyethylene powder and mixed it into the soil uniformly. Then we planted the 3 seeds in each pot at an equal distance and an equal depth. To maintain a consistent environment, we also used growth lights, and we kept them on for 8 hours and off for 16. We watered the plants every other day for 100 milliliters. So how did we collect the data? In order to collect the data, we measured the average plant height and width, and we measured the longest and shortest root length. We also measured the number of leaves on each plant. We also took note of the plant health. We took note of visual symptoms like necrosis and chlorosis. Finally, we calculated germination rate by dividing the number of seeds and the total number of seeds planted.
What is our fact sheet? Our fact sheet just shows what the sample size of our things were, as well as what were the different types of variables used in our experiment.
We had originally seeded 36 plants. However, only 29 sprouted. Now, this impacted how accurate our data could be collected because we did not have the appropriate amount of sample size. So what did we collect?
These three are simple tables that show us the data. As you can see, the lettuce generated a high amount at 1% as compared to the other control groups and experimental groups, whereas for mustard and radish, the experimental groups demonstrated lesser growth than the control. These are the visual representation which I will be explaining more clearly. As you can see, the blue line represents the control group, whereas the yellow, red, and green represent the experimental groups. Overall, for mustard, the control group demonstrated the largest amount of growth as compared to the experimental groups, whereas the 0.5% as well as 1% demonstrated significant growth reductions. Surprisingly, though, the 5% did maintain a constant growth rate as proportionate to the control. For the lettuce data is the plot twist. As you can see, the blue line represents the control, whereas the other line colors represent the experimental groups. As you can see, the 1% demonstrated a larger growth rate than the control, showing that under increased concentrations of the polyethylene microplastic, the lettuce plant did demonstrate a larger growth. The radish plant demonstrated a similar pattern to the mustard, in which the radish plant experimental groups were less than that of the control. However, the 5% did quite well and was touching or even greater than the control groups in the earlier stages, though this advantage did diminish in later stages. This is the submetrics of our project, which are the germination rate, average number of leaves, and the different root lengths. As you can see, the radish demonstrated the best germination rate, whereas mustard did not do as well and demonstrated the lowest germination rate. The average number of leaves stayed the same for all plants, whereas the radish demonstrated the largest root lengths as compared to the other plants, showing that radish took in more microplastics.
So what did our data tell us?
From the observed data and graph analysis, this experiment measured the effects of microplastic concentrations ranging from 0% to 5% on the growths of mustard, lettuce, and radish, specifically Brassica rapa, Lactusa sativa, and Rasphanus sativus over 19 days. The key metrics observed were average plant height, width, and visual changes in sprout appearance, while the submetrics were root length, germination rate, and number of leaves. In general, the control group exhibited the most consistent and significant growth in height, width, and germination rate, while plants grown in soil containing higher concentrations of microplastics did demonstrate stunted growth and irregular sprouting. Specifically, the mustard and radish plants showed significant growth reduction at 1% microplastic concentrations, whereas showed a lower germination rate, reduced height, and thinner stems. The lettuce plants showed less pronounced but still noticeable negative effects with variation in sprout height and a smaller final width, leading to irregular growth patterns. Overall, higher microplastic concentrations do tend to negatively affect plant growth. However, exceptions were observed. For the mustard plants, the 0.5% and 5% concentrations had slower initial growth. However, they showed steady post germination in the later stages, and even though the final growth was still slower than the control. For the lettuce plants, the 1% concentration had a similar or slightly greater final height and width than the control and demonstrated a faster growth rate. As for the radish plants, the 5% concentrations showed higher growth rates at certain times in both height and width.
Now there are two ways to measure growth rate. The first being growth rate formula and the second being slope.
The growth rate formula is a mathematical way of showing the growth rate of the different plants. As you can see, for the lettuce plant, the 1% polyethylene concentration demonstrated a larger growth rate of 0.201 as compared to the control group with a growth rate of 0.178, even though the control group demonstrated the highest width was 0.105. For the mustard plants, the control group did demonstrate the highest growth rate, whereas the 1% and 5% did do better than the 0.5%. And finally, for the radish, the growth rate was highest, around the control group, as you can see for both the height and width, showing that mustard and radish did demonstrate similar patterns, whereas lettuce is the outlier.
Now, the second way to measure growth rate is slope. As you can see, in the average height and average width of mustard, the highest growth rate was on the 1%. As for average width, the highest growth rate was on the control. As for lettuce, the highest growth rate was both on the control, which was interesting compared to the mustard.
Even the 1% for lettuce did show a higher growth rate.
Now, the average growth rate of radish was relatively similar to lettuce with the highest as for control. So, what are t-tests? T-tests determine if there is a significant difference between the means of two groups of datasets. T-tests show whether results are statistically significant or due to chance. We used paired t-tests to compare the average plant height and width in control versus the experimental groups after microplastic exposure. So what did our t-test show us? The paired t-test showed that in average height, no statistically significant difference for any plant types across all microplastic levels. For width, the radish and lettuce also had no significant difference. However, mustard was statistically significant. So what did we conclude? Mustard was the only plant affected, and we suspected that other factors may have influenced our results. So what do these two graphs tell us? These graphs are a side-by-side comparison of the average height and width throughout all of the plants. This makes it easy to understand what our t-tests show. So what was our bias? The tests indicated a use of simulation, and more factors may have contributed to the experiment. Therefore, the margin of error is higher. Some data may not have been proper in the case of usage, as some appear as conflicting. However, through further testing in real-world environments, this simple problem can be solved.
Using tests on microplastics' effects on high photosynthesizing plants, this study does provide a comprehensive insight into how different factors interact with each other. They aim to set a start for more research about plants, environmental factors, and microplastics, advancing the field of environmental technologies.
So in conclusion, this study investigates the effects of polyethylene on plant growth, focusing on the plant's mustard, radish, and lettuce. In general, the results showed that increased microplastic concentrations led to stunted growth, reduced germination rates, abnormal sprouting in all three plant types, with mustard and radishing experiencing the most significant decline at the highest microplastic concentration.
The findings suggest that the adverse effects of plant growth increased as microplastic concentrations increased. These factors do correlate with the plant's phenotype, which was also observed and recorded, which was measuring the amount of growth the plants exhibited daily and recording each of the metrics. Research should investigate the interaction between microplastics and diverse plant types, including their impact on soil structure, fertility, and water retention, like the different microbes which may impact.
And here are our references.
Do you have any questions?
If so, email us. Thank you so much for watching.